Welcome, everyone, and thank you for virtually joining us for the latest version of the Variant Effects seminar series. My name is Diego, and I'm a member of the VES Organizing Committee, along with Stephen Airwood and Mireia Soma. And I'm super excited to introduce two new members of the Organizing Committee team, Adrini D'Souza and Jan Ilbudu, who you'll be seeing more of in the future. These seminars are made possible with support and a lot of help from Laura Muffley and Alice Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. As a reminder, we are always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I just have some housekeeping items to mention. Each talk is about 20 to 25 minutes, so there's time for some extra questions. Today, there's just one speaker, so there's definitely time. Please post those questions at any time to the Zoom Q&A, and we'll read them out to the speaker at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can catch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. Don't worry about copying this whole URL. I'm going to post this link and the others that I mentioned to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. In our upcoming December seminar, Megan Lay from the University of Texas, Austin, is going to present her work on tracking 10,000 years of natural selection by studying DNA from 1,000 ancient genomes. So be sure to save that on your calendar. And then for the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. Without further ado, we have a great speaker scheduled for today's presentation. Dr. Tyler Starr is a K99 fellow and incoming assistant professor in the Department of Biochemistry at the University of Utah. His research explores the evolutionary arms races between viruses and host factors involved in infection and immunity. Since 2020, Dr. Starr has applied high-throughput deep mutational scanning assays to study SARS-CoV-2, informing ongoing efforts in viral forecasting and aiding in the development of vaccines and monoclonal antibodies to combat COVID-19. The title of his talk today is Evolution of SARS-CoV-2 and Related Bat Coronaviruses. Tyler, feel free to start sharing your screen and go ahead and start whenever you are ready.
Awesome. All right, thanks for the introduction. Thanks for everyone who's attending. And I'm excited to talk to you all here about some of my recent work on evolution of SARS-CoV-2. And then at the end, we'll zoom out a little bit more to understand some of the deeper evolutionary events in bat coronaviruses. So just kind of starting big picture, my research interest is in studying the evolution of proteins at the host virus interface, just because this is, I think, a place where there's a lot of really interesting protein-protein interactions that drive these rapid dynamics of evolutionary change. And so the work I've done thus far is really centered on these viral surface proteins, these spike glycoproteins that decorate virus surfaces and have kind of two primary competing sorts of arm races that drive really interesting evolution of these proteins. The first is sort of arms race with reservoir receptors, where a lot of organisms that host these viruses natively have evolutionary arms races to diversify their receptors to avoid viral entry and kind of counteracting viral evolution to kind of keep up with receptor diversification. And on the flip side, hosts are also developing antibody and other immune responses that tend to target these spike proteins, which drive these dynamics of evolutionary chase and escape to avoid antibody neutralization and recognition. And so in the work today on coronaviruses, this glycoprotein that we're kind of thinking a lot about is the spike protein, which I think many of us are familiar with. And my research in particular has focused on a particular domain of spike called the receptor binding domain, or the RBD. And this is this kind of autonomously folding domain that sits at the spike trimer apex. And it's responsible for encoding high affinity binding to ACE2 receptors on host target cells. And so it's this high affinity interaction between RBD and ACE2 receptor that enables the initiation of cellular infection via fusion of viral and host membranes. So because of this key role of the RBD in receptor binding, there's a lot of key evolution in this domain that's been occurring over the short-term SARS-CoV-2 pandemic and over longer-term coronavirus evolution. So for example, it's been well described in prior examples how evolution of RBD is really important to the ability of these viruses to cross species boundaries and be able to jump to using different ACE2s on different hosts and different spillover hosts. And the RBD is also the dominant target of neutralizing antibody immunity. This includes things like monoclonal antibodies that have been used as therapeutics over the course of the SARS-CoV-2 pandemic. It's also the dominant target of immunity in the types of vaccine responses and the convalescent patient serum responses we've all been able to develop through the course of vaccination and infection. And so it's kind of corresponding with these key roles of the RBD. We've seen diversification of SARS-CoV-2 over time through these different variants of concern, which include key mutations in the RBD that are really central to some of the important phenotypes of these variants of concern that we'll talk more about. But the RBD is also one of the most variable parts of this viral genome over kind of longer term evolution of these so-called SARS-related coronaviruses or sarbecoviruses. And so here I'm showing sort of phylogeny of all the bat viruses that are known, where we see SARS-CoV-2 and SARS-CoV-1. And sort of over this longer evolutionary timescale, again, this RBD is under kind of rapid evolutionary pressures. And so this is probably not a tough pull for this audience, but kind of in both of these regimes of short-term SARS-CoV-2 evolution, where we've had like billions of genomes kind of coming in worldwide, as well as kind of the more sparse sampling of natural diversity of these viruses in nature, our ability to see genetic variation through advances in sequencing obviously outpaces our capacity to evaluate the functional consequences of this genetic variation. And so half the time when we're sampling new SARS-CoV-2 variants or new sarbecoviruses in nature, we see the genetic variation, but we don't know what it means. And so the work we've been doing has been intending to use these high throughput biochemical measurements to kind of better aid this effort in evaluating what genetic variation means. And so to do these high throughput biochemical measurements, we've been leveraging a deep mutational scanning platform on the basis of yeast surface display. So yeast surface display is sort of a classic method in many aspects of protein engineering and deep mutational scanning. And so what we're doing is taking yeast and having them express this folded spike receptor binding domain, or RBD, on the surface of individual yeast cells. And when we have that RBD on the surface of yeast, we can probe different biochemical properties of the yeast expressed RBD via different fluorescence couplings. For example, we can use sort of a C-terminal tag and an antibody that detects that tag to measure how efficiently different RBD variants are expressed on the yeast cell surface, which is sort of a proxy for folding stability. We can do things like incubate with soluble ACE2 receptors from human or other relevant species and detect whether these displayed RBDs can bind to these different ACE2s. Or we can also incubate with monoclonal antibodies or polyclonal sera to understand how different RBD variants impact antibody binding. And so, yeast display is often done in sort of an isogenic context where you just have a population that's expressing a single RBD and you can measure these different properties. Of course, we want to do this in high throughput via mutational scanning approaches. And so the way we do this is to create, of course, variant libraries. So yeast populations where each cell has a different RBD variant on their cell surface. And I'll talk about a couple different ways we've done this, but we can do this, for example, with amino acid mutants of a focal strain of interest, or in the latter half of the talk, I'll talk about how we can use this approach also to query like a panel of different homologs across the diversity of these SARS-related coronaviruses. What we then do is take this library, incubate for one of these properties of interest to get a fluorescence signal. And then we use fluorescence activated cell sorting, or FACS, to basically partition this mixed library into like low fluorescence and high fluorescence bins, kind of separating which variants in the library have one of these activities or not. We then use quantitative deep sequencing to resolve which cells are in the low bin or in the high bin. And then depending how we set up the experiment, for example, if we do this process across multiple ACE2 concentrations, we can actually fit these actual titration binding curves where we get sort of a binding curve and a dissociation constant like metric for each variant in our library simultaneously, giving us these really nice kind of pseudo-biochemical measurements in one single massively parallel experiment. So today I was going to talk hopefully about three kind of different ways that we've used this platform to study the evolution of SARS-CoV-2 and these related bat sarbecoviruses. So I'll first talk about some of our initial studies kind of at the beginning of the SARS-CoV-2 pandemic, where through these types of mutational scans, we described this tolerance to mutation within this critical domain of this virus and what we've seen that's meant for its future evolution. I'll then talk about some more recent work where we've been studying the role of epistasis in kind of shifting these mutational landscapes and how that might impact future SARS-CoV-2 evolution. And then we'll zoom out to think more broadly about how these properties of ACE2 binding are evolving across deeper sarbecovirus evolution and how that might relate to zoonotic potential. All right, so this first project takes us back to like the beginning of the pandemic. And at that point, I, like many other people, were just reading everything we could about coronaviruses. And it was pretty clear that these receptor binding domains were going to be important to this virus and its immunology, but we really didn't know what its capacity for future evolution was going to be. And there was actually a common thought at that early time that there wasn't going to be much variation and evolution in this virus. And so to kind of begin to shed light on this question, we decided to just do a simple demutational scan of the SARS-CoV-2 RBD using the SEAS display platform. So we're taking each of the 201 sites in this pretty small domain and measuring the effect of each of the 19 possible amino acid mutations that can be introduced. And so when we do that, we get a heat map like this. I assume many of us are familiar with these heat maps, but just to kind of go over them, each of these columns is one of the positions in the RBD. We have the 20 different amino acids that we introduce, and then we color them according to the scale bar where we are doing these titrations to get these dissociation constant-like metrics, where blue are mutations that enhance affinity for the human ACE2, white are mutations that are just neutral with respect to this phenotype, and then darker colors of red are those that are increasingly deleterious for ACE2 binding. And so kind of again, of no surprise to this audience, we see a lot of heterogeneity across sites. So there's plenty of sites like this one here, 488, that are entirely constrained. All these mutations are dark red. So this is a site that's probably not going to vary over the course of viral evolution. But what was more surprising to us were all these positions that look like this one here, where even though this is actually a known ACE2 contact residue that's thought to be really important for ACE2 interaction, we see tons of heterogeneity in the effects of mutations at this site. There's plenty of mutations that enhance affinity or are more or less neutral, as well as some that decrease affinity. So when we generated this map, this was before any sort of variation in SARS-CoV-2 had really started to accumulate. But we looked at this and thought, there's a lot of tolerance to mutation in this domain and even perhaps some evolvability. And so, maybe it was not too much of a surprise starting in kind of fall of 2020 when the first alpha variant emerged and subsequent variants began to convergently fix mutations that from these maps like this one, we thought looked of potential interest. And so what I'm circling here is a mutation N501Y, which has been kind of central to the emergence of several variants of concern. And our maps showed was an affinity enhancing mutation. And in kind of the middle part of the talk, I'll talk a lot about what I think that affinity enhancement is really important for the evolution of this virus. A couple other things I'd like to point out is that mutations like N501Y are not really unique. There's plenty of affinity enhancing mutations in this map, suggesting that the potential for evolution of this virus is not likely to be exhausted. But kind of the other pattern I think is less obvious at first, but I think many of us would appreciate is extremely important, is all of this kind of white space, all of this neutral variation with respect to ACE2 binding affinity. And that's because this is the space of mutations that the virus can sample more or less freely with respect to this overarching constraint of receptor binding while it tries to evolve in other ways. And so I'll transition to kind of a small part of this talk where I talk about what is one of those really important ways that this virus is evolving. And that has to do with how the virus needs to evolve to escape antibodies. And so I was going to go over kind of a couple vignettes about some of these monoclonal antibodies that were being developed as therapeutics. And I think they nicely show kind of how efficiently this virus can kind of weasel around immunity and render things like monoclonal antibodies ineffective. So when different groups at the beginning of the pandemic started trying to develop monoclonal antibodies, a central idea was to prioritize antibodies that contact whose epitopes on the receptor binding domain sort of overlap with its ACE2 contact sites. And the thought behind that was ACE2 contact sites, those should be the constrained positions. So if we target those with antibodies, the virus should have reduced capacity to escape from these antibodies. And when we looked at our demutational scanning map, we could see, if we take our average mutational effect on ACE2 binding and map it to the structure, and then show spheres, the actual ACE2 contact sites, we can see that indeed there are some very constrained ACE2 surface or ACE2 contacting sites, but at the same time, there's plenty of tolerable positions within this surface as well. So it's more of a mosaic of constraint and tolerance. And what we saw was that when some of these early studies, for example, the scientists from Regeneron started seeing, what is the capacity for evolutionary escape from the monoclonal antibodies they were developing, they might grow replicative virus in the presence of their antibody and sequence the variants that come out. We could compare these to our maps and show that really efficiently the virus was taking this kind of mosaic surface and really finding the tolerated mutations within this surface that enable escape from antibody binding without reductions in ACE2 binding, really showing how both these kind of biochemical properties we can measure, like ACE2 binding, really do have relevance to what the virus is going to do. And so to address this a little more head on, we started using this platform not only to measure ACE2 binding, but also to measure binding by these monoclonal antibodies to understand the breadth of avenues of escape from binding by different groups of antibodies. And so what I'm showing here is one of these mutational scans we did for escape for binding from, again, one of these Regeneron monoclonal antibodies that was one of the early monoclonals approved for use in the clinic. And so what I show here is basically the 200 sites in the RBD, and then our metric of antibody escape on the y-axis, which shows these kind of peaks, which reflect the positions where mutations enable escape from binding by this antibody. We can zoom in on those peaks and see the individual amino acid changes that enable escape at each of these positions. And then we can also do things like layer on our ACE2 binding scores, such that mutations that are in dark brown are those that are most neutral with respect to ACE2 binding, whereas those that fade away are the ones that exhibit more constraint and would have more of a deficit of ACE2 binding. And so for this antibody, as an example, you can see there's variation. Some escape mutations don't suffer much deficit, whereas others are much more constrained. And even though they escape antibody binding, would probably have other fitness deficits. Kind of consistent with that, if we show here in squares, these are the mutations that came up in that study I showed in the last slide, again, showing that they're kind of sampling the more dark brown tolerated mutations within this heterogeneous landscape. I'll just show really quickly, we can also map these to the structure and kind of show the sort of functional footprint of escape that of course maps to where the antibody binds, but it gives us a little bit more the kind of hotspot understanding of where are the key energetics for interaction. For example, this phenylalanine really jumps out as being really focal in this larger structural interface. And so I'm not going to go too much into a lot of the different antibody studies that we've done, but just to kind of emphasize one key result from our studies was that, what we saw again and again was that there was no shortage of paths of escape from antibody binding. So here's another antibody used in the clinic, LY-CoV-555, and we can see, our map that we get. And kind of correspondingly, or sort of unfortunately for the usage of these antibodies, a lot of the positions that are in these monoclonal antibody footprints were also those that were being driven to evolve in variants of concern, such as the beta and gamma early variants or Omicron, or other mutations like at 452 that are present in Delta and Eta. And so a second approach that was used to kind of counteract viral evolution was just to simply put two antibodies together in a cocktail. And so when this antibody started to become less effective because of some of that early evolution, they just started throwing it in as a cocktail with another antibody. But again, kind of unfortunate because of the evolutionary potential of this virus. A lot of these variants also fixed kind of combinations of mutations that were present in each escape map, thereby escaping even the cocktail, which is often the kind of standard approach to stymieing viral escape. Or in one case, we even found a single mutation that can escape binding from both antibodies simultaneously that later emerged in Omicron. And so I won't go into some more of the work we've done in this direction, but really one thing that's emphasized to us was that, all the effort that goes into monoclonal antibody development can get rendered really ineffective really quickly if you end up with antibodies that are easily escapable. And so we think it's really important that some of these ideas about constraint of epitopes and escapability, which can be aided a lot by DMS, really might be very useful to incorporate into really the early stage of antibody and cocktail design, because we find ourselves at this point right now in time where basically there's no approved authorized monoclonal antibodies because they've all been escaped thus far by ongoing virus evolution. All right, so I'm going to shift gears a little bit now. That was all based off of kind of our early D mutational scanning libraries in sort of the ancestral strain of SARS-CoV-2. But as I started to kind of highlight with some of these variants of concern, some of the backgrounds of the RBD itself have been changing over time. And so, as I said, the Wuhan 1 strain, that's where we did our first DMS. But there's been kind of increasing numbers of mutations accumulating in these variant of concern RBDs. And so when we have this map of ACE2 binding effects, for example, in the ancestral strain, really our next question is, okay, if we start to fix mutations in this map, how does this mutational landscape itself change? And so this is a question about epistasis, which is this concept of the non-additivity of mutational effects, or as I'll talk about a lot today, epistasis also results in the same mutation having different effects on different sequence backgrounds. And we know that over at least long-term SARS-related coronavirus evolution, that epistasis can be really profound. So we had this one example here with the same N501Y mutation, where in SARS-CoV-2, we know this is an affinity-enhancing mutation that's been really important for the development of this pandemic. And so in this binding curve, we can see that shift to the left, reflecting the affinity-enhancing nature of that N501Y mutation. In a prior study, we had introduced this homologous mutation in other sarbecoviruses like SARS-CoV-1 or other more divergent sarbecoviruses. And most of the time, we actually saw this homologous mutation was strongly affinity decreasing. And so this mutation that's so important to SARS-CoV-2, because of epistasis, never really would have been predicted to have been important from studies in other sarbecoviruses, really showing how, at least over longer term evolution, these two RBDs differ at 25% of their amino acid positions. Over that larger divergence, this epistasis can be really important. What we wanted to understand was, over this sort of shorter divergence of some of these variants of concern that fix, one or two, sorry, my Outlook is popping up with something, whether there's, again, some evidence of this epistasis and whether it matters for the virus's evolution. And so we repeated these deep mutational scans as before in the Wuhan 1 background, but now we just fixed substitutions that define some of these variants of concern and repeat the process. So measure all the mutational effects, but now when we fix first this N501Y substitution. And I won't really talk about it today, but more recently in a preprint that should come out soon in PLOS Pathogens, we've also done this in some of the newer Omicron variants as well. But really at this high level, obviously heat maps are sort of hard to digest, but you can begin to see some trends, such as mutations that are blue in one background and white in the other, or regions that are darker red in one background and maybe more tolerable or pink in another. But to kind of more efficiently kind of visualize where these epistatic shifts and mutational effects are occurring, we derived this epistatic shift metric, which is kind of a probabilistic-based measurement of the divergence of site-specific mutational profiles. And so to kind of calibrate that, I'm showing here the two extremes, positions where there's little epistatic shift and the position where we saw the most epistatic shift. And to kind of help understand what this means, I'll explain these plots. What I'm showing here on the y-axis is the affinity of a measured amino acid in the beta background. So one of these variants of concern that fixes 3 substitutions versus the x-axis, the affinity of a measurement in the Wuhan 1 ancestral strain background. And so in a position like this, site 484 or many other sites in the RBD, we see little epistatic shift, which can be reflected in the fact that these 20 amino acid letters, the measured affinities of the 20 amino acids, are really on this 1-to-1 line. There's no change in the effect of different mutations at site 484 from one background to the next. And that's in contrast to site 498 here, where we see a lot of variation off of this line, reflecting mutations that have increasingly different effects in one background or the other. So that can be mutations that are down here in this quadrant that are affinity enhancing in the ancestral background, but now slightly decrease affinity in beta, or vice versa, mutations that strongly enhance affinity in beta, but are actually slightly affinity decreasing in Wuhan 1. And so we can take this epistatic shift metric and just plot it across each site in the genome or in the RBD as a sort of line plot. And so I'm showing here 2 variants, delta and eta, where we didn't really see very strong epistatic shifts when we compare that to the alpha and beta variant, where we see all of these blue peaks reflecting sites where there's these strong perturbations of mutational effects, again, with 498 being that example I showed before. And really what's shared between alpha and beta that explains their kind of overlapping peaks here is the fact that they both have this N501Y substitution that I keep talking about, which really seems to be a central kind of epistatic determinant or perturber of this mutant landscape. When we map these epistatic shifts to kind of the structural backbone, we can see that these epistatic shifts include, so this is N501 here, we can see that the strongest epistatic shift at site 498 is the direct contact residue of N501, but we can see that there's also kind of second shell kind of knock-on effects of these epistatic perturbations where, this loop back here doesn't directly contact N501, but gets a lot of large shifts, or it can percolate down this strand here, or down this kind of set of residues here. And so structurally, it makes sense why these sites are being perturbed. Although I think it's really interesting to note that you couldn't necessarily predict or easily imagine that there were going to be these epistatic shifts from structures alone. So what I'm showing here are structural alignments of RBD structures that have been determined in each of these variants. And really the point here is that the backbones of these RBDs are completely identical among these 4 different backgrounds, despite the fact that these 2 have a lot of these epistatic shifts. So it's not accompanied by some large-scale structural change that would immediately clue us into the fact that mutational effects are going to differ. And that's true even if we zoom into individual side chains. So I'm showing here in blue and gray the beta and Wuhan 1 variants, multiple structures of each. We can see the N501Y change here, And site 498 is the one that I said was the strongest shift. But if we look at these overlapping structures, Q498 has a lot of heterogeneity from structure to structure, but it's not like different in the betas compared to the ancestral strains, and the contacts aren't seemingly different. And so there's not anything like statically that's going on, but instead there must be something more dynamic about the interaction that changes due to N501Y to cause all of these shifts in mutational effects, which I think is extremely interesting. I'll next kind of go over one example of an epistatic shift that we have now seen is of very high consequence for SARS-CoV-2 evolution. And this is this interaction between N501Y and its potentiation of this Q498R mutation. And so to kind of visualize this epistasis, I'm showing here like a mutant cycle diagram where I'm showing the effect of either these single mutations as measured in the Wuhan 1 background. And so if there were no epistasis between these mutations, we'd expect the double mutant to have an affinity right here. This should just be like a parallelogram shape. But instead, we see that this double mutant has a massively enhanced binding affinity, way higher binding affinity to predict from these effects of the single mutants alone. And so when we first saw this, we measured this data set, I think in like October of last year, so about a year ago. And we saw this extreme interaction, but we didn't see any evidence that this had mattered for SARS-CoV-2 evolution until we fast forwarded about a month and Omicron emerged, fixing this combination of 2 mutations along with 13 other changes in its RBD. And what we've been able to see, or what we kind of think is going on with these 2 mutations, is that they have a really important role in basically buffering affinity such that you can then enable large degrees of viral antigenic evolution. And so to kind of show why we think that's the case, what I'm doing here is taking the 15 changes in the Omicron RBD and kind of just introducing them in order of their rank order effects on affinity, not saying that that's the order they occurred on evolutionarily, but just to kind of visualize. So you can see that these 2 mutations, because of their epistatic interaction, give this large affinity boost, which then is kind of added on by a couple other smaller mutations. And then some of these other mutations in Omicron have these like small little deficits in ACE2 binding affinity, such that when you sum all 15 mutations together, you end up with a predicted affinity that's basically the same as the ancestral strain. And in reality, the measured Omicron affinity is something, it's within 2-fold or so binding affinity compared to the ancestral strain. And so, the question is like, what is this doing and what are these mutations doing? So what I'm doing here is taking these same 15 mutations, but now plotting them on like a 2D scatter plot where the y-axis is their effect on binding affinity, so positive affinity buffering mutations and affinity decreasing mutations. And on the x-axis is this sort of composite antibody escape score that we can determine from the large number of sort of antibody escape experiments that have now been performed by us and now in higher throughput by other groups as well, where we can kind of estimate the expected impact of different mutations on polyclonal immune response. And so what we can see here is that the mutations like Q498R, N501Y that are enhancing affinity, these really aren't antigenic escape mutations. They have hardly any antibody escape phenotype, whereas a lot of these antibody escape mutations have these sort of small ACE2 binding deficits. And so we think what's happening is that you need larger and larger affinity buffers to enable more and more antigenic mutations, more and more mutations with different epitopes that allow you to escape further and further away from polyclonal immunity. We think this might be more general with just Omicron. We can look back at some of the earlier variants like Beta and see, they got a smaller affinity buffer, and so they just couldn't evolve as far antigenically. And so perhaps this extra synergy, this extra epistasis here that gave this extra large affinity gain enabled even further antigenic changes, leading to the extreme Omicron phenotype that enabled such dramatic reinfection and evasion of Wuhan 1 strain immunity. All right, so I'll go over kind of one more thing about this epistatic shifts that we think is really important, which is that, I showed one example that was of very focal interest for Omicron emergence, but we think we see evidence in the SARS-CoV-2 evolution, sequence evolution, that these epistatic shifts are mattering on a larger scale. So what we did here was take a global phylogeny of all of the SARS-CoV-2 sequences that are assembled in this great Usher Matt Utils tree. And basically just count the occurrence of substitutions and partition as to whether they occurred on the backdrop of an N501 genome versus the Y501 genome. And so that's what we're showing here, are mutational occurrences over SARS-CoV-2 evolution. To the right of this line are mutations that occur more frequently on the background of Y501, and to the left are mutations that occur more frequently on the ancestral N501 state. And the y-axis are measured delta-delta binding constants, so mutations that have better effects on affinity on Y501 versus better effects on N501. And so we can see that the mutations that are most enriched for our epistatic measurements of binding are indeed occurring more frequently on the backgrounds of those genomes. So this goes beyond Q498R to explain some of these other mutations at other antigenically important sites, kind of suggesting that, as Y501 genomes predominate, now that Omicron has basically taken over, some of these sites that might not have been evolving very rapidly or very convergently in the early pandemic might be becoming more relevant in future SARS-CoV-2 evolution. All right. So to keep kind of zooming along here, I'm going to jump now to this kind of last story where instead of just thinking about how sequence evolution is happening over kind of short-term SARS-CoV-2 evolution and how these large-scale mutational assays can inform that, I'm now thinking over kind of deeper evolutionary time as these viruses circulate in bats and acquire traits that allow spillover into humans and other species, and how we can think about what's going on there to understand, not yet, but what I will eventually want to get to is a better understanding of what are these molecular evolutionary forces that drive these properties of viruses that enable things like human spillover. And so, again, this is focusing here on these SARS-related coronaviruses, which are this larger family of viruses from which the human viruses, SARS-CoV-1 and SARS-CoV-2, have transiently spilled over, or I guess in the case of SARS-CoV-2, not so transiently. But again, this is a larger clade of viruses where there's many kind of closely related bat and intermediate species or other spillover species viruses that are closely related to the human strains. And these are all kind of found in rhinolophus bats in East and Southeast Asia. But there's also these other two clades of these sarbecoviruses where spillover has not yet been observed. So this includes this orange clade down here, which are also in many of these rhinolophus species from Asia. And then it also includes this pink clade up here, which are found in rhinolophus bats in Africa and Europe, for which at the beginning of this project, there were really only two known representatives. So these sarbecoviruses I kind of started with in the introduction have a lot of genetic variation. So this is kind of percent identity to SARS-CoV-2. And you can see in this most divergent clade, they're differing at almost 40% of their amino acid positions. And prior to us doing this project, there was also kind of unclear variation in ACE2 binding across these clades. So a lot of viruses from the SARS-2 clade and SARS-1 clade had been shown to be able to bind to ACE2 from humans and in some cases civets or bat species. But as far as it had been tested, no RBD from this orange clade nor in Africa and Europe had been shown to bind to any species of ACE2 ortholog. And so kind of with this prior state of knowledge, there were kind of two different parsimonious evolutionary histories that we could draw on this tree to kind of think about how was ACE2 binding evolving over time. And really the key is that in either of these scenarios, we would hypothesize that the ancestor did not bind to ACE2. That's because these two non-ACE2 binding clades are kind of separated on the tree. And we would posit that the gain in ACE2 binding was somewhere later in the evolution of these viruses. And so we wanted to kind of address these histories a little bit more rigorously or a little bit more comprehensively rather than the more so sporadic tests of ACE2 binding that have been conducted thus far. And so we used our yeast display platform to basically clone in each of these RBDs across the 45 or 50 or so viruses and measure binding to different orthologs of ACE2. So I'm showing here first our human ACE2 binding data, where I'm showing as a heat map where darker blue indicates higher binding affinity of these different RBDs. And so kind of consistent with what had been shown in the most exhaustive studies to date is that, we got, high affinity of human ACE2 binding in the human viruses and a lot of their close animal relatives. But again, we saw no binding in Africa, European sarbecoviruses, nor in this orange clade. Kind of just fast forward all the data. We repeated this process with different ACE2s from sort of other spillover species, as well as a couple of alleles from the types of rhinolophus bats that harbor some of these sarbecoviruses. And really what was most interesting and what I really want to focus on was that we detected binding to some of these rhinolophus bat ACE2s in this bat KY72 RBD from Kenya. So this was the first time that any sort of ACE2 binding had to be detected in one of these viruses from outside of Asia. And so we wanted to validate this. And so I worked with Sam Zapita, a graduate student in David Wiesler's lab at University of Washington, who constructed these sort of pseudotyped viruses that leverage the BAT KY72 full spike and asked whether that could enable entry into cells that are expressing these rhinolophus affinis ACE2s. And indeed, she saw kind of consistent with the strength of binding we saw to these two different alleles that they could promote entry of this spike, suggesting that, even though this isn't from a rhinolophus affinis bat, we suspect it might be using the ACE2 of its matched rhinolophus bat species, and that it is truly an ACE2 utilizing virus. And the reason I think this is interesting is because, detecting binding to ACE2 in one of these viruses out here might kind of push back our expectation of when ACE2 binding originated in this family to be at the, the ancestral point of Sarbecovirus evolution, with just human ACE2 binding being the kind of variation that's emerged a little bit more recently. And so this is kind of our updated parsimonious reconstruction. To gain more insight into it, we also leveraged ancestral sequence reconstruction to hypothesize plausible sequences that could represent these ancestral nodes and then test their binding affinities. And again, kind of consistent with this trajectory here, we found that the Sarbecovirus sequence that we reconstructed could indeed bind to bat ACE2s, and that ants Asia is when the human ACE2 binding and some of this more promiscuous ACE2 binding evolves, and that it is indeed a derived loss of ACE2 binding that's present in this orange clade, the so-called clade 2. All right, so that's kind of the first part, which is where we established that ACE2 binding seems to actually be an ancestral trait of Sarbecoviruses with sort of broader geographic and phylogenetic breadth than had been previously known. And that human ACE2 binding still did evolve, long before SARS-CoV-1 and SARS-CoV-2 zoonosis, and it's present in a lot of these animal viruses. What we next wanted to do was use some of our kind of mutational approaches to understand not just the natural variation, but also what's the evolutionary potential of these domains. And so from our prior demutational scan, when we kind of zoom in at the sites that are at the RBD interface, we picked 6 positions that looked particularly interesting or particularly evolvable from our prior study or from other studies. And so these are the 6 positions we chose to do this sort of a mini mutational scan where we introduce each of the 19 amino acid mutations at each of these 6 positions, but do this tiled across several different Sarbecovirus backgrounds. So I'm showing here the different Sarbecoviruses that we did this kind of mini mutational scanning in. Each of these red lines is sort of the parental affinity of this RBD for human ACE2 in this case. And then each of these dots is one of the mutations that we tested. And so there was a lot of interesting stuff that jumped out. First of all was that, regardless of the parental binding affinity of each of these RBDs for human ACE2 in this case, we could always find mutations that could make that binding stronger. And furthermore, in a couple of these backgrounds where there wasn't kind of a parental binding affinity, nonetheless, we could identify individual mutations that conferred this binding affinity, really suggesting these types of viruses like the ancestral Sarbecovirus or there's bat KY72 virus from Africa aren't really far away from ACE2 binding. They have high evolutionary potential. And I'll just skip over. We validated some of that with more typical biochemistry binding assays and pseudoviral assays. When we kind of just throw up all the data for all the different ACE2s, we see this as in particular to the human ACE2, kind of in general, there's always potential for evolution of binding to different ACE2s, even those that might not be natively used by different viruses, really suggesting that this RBD is very mutationally pliable and very evolvable to be able to adapt to different species ACE2 really easily. And that's probably really central to the kind of porous species boundaries that we see in these viruses. All right, so for my last sort of slide, I kind of want to end with a so what. So why should you care so much that I'm throwing this conclusion down your throat that ACE2 binding is ancestral and evolvable? And so we see that this kind of binding to ACE2 and the evolvability of human ACE2 is a trait that's present even in ancestral Sarbecovirus. So we found that this reconstructed node that we tested can bind to bat ACE2s and is only single mutations away from human ACE2 binding. And kind of repolarizing this as an ancestral trait of all of these Sarbecoviruses means that that's actually kind of the default or assumed state of any Sarbecovirus that might be described out there soon in nature or in viruses that haven't yet been described. So undescribed and newly described Sarbecoviruses likely share this property of binding to bat ACE2s and easily being able to evolve human ACE2 binding unless they happen to cluster with this derived trait. So really kind of emphasizing that this loss of ACE2 binding that was thought to be the native ancestral trait is actually the more kind of out there derived trait that's more particular. And so to really emphasize the importance of this conclusion, kind of while we were doing this work, more Sarbecoviruses were being described from across Asia, as well as a lot more viruses in this critical Africa and Europe clade that we found such interesting details in. And so this includes this RSYN04 virus, which I'm showing in red here because it's actually branching off here independently from the kind of the 4 previous clades that we were talking about. And so this is kind of a nice test of, okay, this is a virus we didn't know, a group we didn't know existed out there. Does it share this trait? And so indeed, when we just tested its binding, we could see that it could bind to bat ACE2, and with just a single candidate mutation from our prior studies, we could see human ACE2 binding was not far away for this virus. Where things got extremely interesting was when we tested some more of these European and African Sarbecoviruses, in particular these Costa viruses, where Costa 2 here we showed even as an unmutated sort of parental RBD, it actually had independently evolved human ACE2 binding, really showing that this kind of evolutionary potential in this clade sometimes is realized. And so there's probably a lot more going on in this clade than we're aware of. And also people who are doing things like developing pan-sarbecovirus therapeutics for future pandemic prevention really shouldn't be ignoring this clade, nor this entire phylogenetic gap that links it to the stuff that had previously been of the most kind of focal interest. All right, so with that, I'll just kind of quickly flash up again my conclusions, which is that via these demutational scanning approaches, we've described this large degree of mutational tolerance in the SARS-CoV-2 RBD. I'm really curious. I know a lot of people like to do kind of meta-analyses of different DMS data sets, and it's always hard to compare from technique to technique. So in general, I'm really interested in doing this kind of yeast display titration for other viral domains to understand whether this is a particularly large degree of mutational tolerance or if this is just how these proteins are. We've seen that this tolerance has been really important, for example, in enabling evolutionary escape from antiviral antibodies, which is what has allowed kind of the ongoing evolution and development of this pandemic. Further contributing to that kind of ongoing evolution is the fact that viral sequence evolution itself can change and expand future routes of evolution, kind of opening up new pathways of sequence evolution, really suggesting there's no dead ends in sequence space, which again is probably not too surprising to any of us. And then last, when we kind of zoom out over a larger time scale, we can see that ACE2 binding, this trait that's so critical for SARS-CoV-1 and SARS-CoV-2 spillover, is maybe more ancestral and evolvable than we had previously appreciated, really informing future efforts in things like viral surveillance and pandemic prevention. All right, so with that, I'll thank my postdoc advisor, Jesse, in whose lab I did all of this work, as well as Ali Greeny, a recently graduated PhD student in his lab, who was fantastic to work with on a lot of this work. A lot of really important collaborators, a couple of people who have started to work with me here in Utah, which is really exciting. And of course, I have to do the shameless plug of, I'm a new PI, and so I'm always looking for people who are interested in this work and these methods and interested in bringing their own perspectives to the types of work we're interested in. This is Salt Lake City. It's really pretty, and we're just up here, kind of the last building before the foothills. And so I have a very nice view from both my office as well as my lab space. So I encourage you to come check it out. All right, thanks so much.
Great, thanks so much, Tyler. That was an incredibly impressive talk. We already have a couple questions coming in from the Q&A. So to get us started, Zhenya Avakine has asked, how conserved is ACE2 across different species? And is there something about ACE2 that facilitates zoonosis? Yeah, that's a really interesting question. So there's not much variation within humans. That's another common question that I get. But among the species, especially the Rhinolophus bat species that harbor these viruses, the ACE2 is extremely divergent. And a lot of that divergence really centers on the residues that these RBDs interact with. And so I think in the ACE2s I showed here, if we look at just RBD contact residues, I think they range from very close identity to human to up to 40% different from human at those, couple dozen direct contact residues. So at the interface, there could be a lot of variation. And I really like the second part of the question, which is how that influences the evolution. I think there's two really crazy things here. First of all is that ACE2 binding has been independently evolved in multiple lineages of coronaviruses. And it's definitely independent because they use different structural motifs, they're completely non-homologous, and they actually bind to different surfaces of ACE2. So I don't know whether it's just its shape or its tissue specificity or something like that, but it's clearly a receptor that these viruses like to keep evolving to use. As it relates to these Sarbecoviruses, a big hypothesis that I want to get at is that because there's so much variation in the bat ACE2s, that the bats that harbor these viruses in nature, there's probably some evolutionary pressure on the viral side to just get promiscuity toward many different looking ACE2s, because these bats are all co-roosting. There's multiple alleles in species, as you can see here, that are all in the same vicinity. So sympatric variation is really high. And so the virus's best way to cope with that is to just not care about that variation, be able to bind to all of it. And probably that's what leads to human ACE2 binding just kind of coming along for the ride. We have a question from Jeannie D'Souza, and they said, I noticed the beginning of the Delta background map is less sensitive to mutation. Could this be associated to the lethality rates of Delta compared to other variants?
I don't know if I would say Delta was less sensitive to mutation. It's more that its mutational map was more similar to Wuhan 1. So it should have the same, overall tolerance to mutation as the Wuhan 1 strain. That epistatic shift metric is not tolerance, it's how different are mutations from their effects as we first measured in Wuhan 1. And so Delta just had the mutations in Delta L452R and T748K just aren't perturbing the map. They kind of point out into space in the RBD. And so they're not like changing the effects of other mutations at other positions.
Great. And so we have a few more questions coming in. So Reed Brewers asked, are there efforts to adapt computational predictors for this type of work? If so, how are they performing? The experimental data is incredible, but as investigations become more numerous, when you're looking at epistasis and new variants of concern, presumably a computational approach might be helpful. So is there any work that you're aware of that is coming along, and how is it doing?
Yeah, so I'm not doing that work because I'm not very good at that stuff. I like to just do the measurements, but I agree it'd be nice if we didn't have to do so many measurements. I know there are groups that have definitely taken our data to do different things with it. I haven't seen for SARS-CoV-2 forecasting. I don't know. It's so hard because so many times with these models, it's like they work until they don't. And it's just hard to understand how they're doing. I'm trying to think if someone's fitted retroactive data to predict beta or Omicron emergence. And I haven't seen it done super effectively. There's definitely a lot of work in that area though. And I think it would be really interesting. But I think it's got to be really hard because the Q498R you couldn't have predicted until you had an N501Y data set. There was just such a specific epistatic interaction that was so strong there. Or if you wanted to apply this to broader Sarbecovirus evolution, as I kind of pointed out, N501Y, in our different maps here, we did 14 different backgrounds across this tree, and only in the couple that were most closely related to SARS-CoV-2 did N501Y start to become affinity enhancing. And so surely there's other examples like that where we just don't have the right background to measure an important effect that's going to be important to a virus's evolution for whatever other clade is out there. So that's just the hardest thing with epistasis, is just it's idiosyncrasy, I guess. But I agree it'd be very important to solve, or very useful to solve.
Right. And I think as you accumulate more and more of these data sets, it might become slightly easier to start developing computational predictions.
Yeah.
So we have a question from Noam Priwis. They asked, how do coronaviruses that don't bind ACE2 vary compared to this clade?
I guess I'm not sure if he means this clade that has lost ACE2 binding. They have a lot of variation. And we just don't know what receptor they're using yet. If you're talking about other deeper branch points from all of the Sarbecoviruses, I think RBD variation is high in any of these coronavirus families. I think the RBD kind of is always at this critical juncture of being receptor binding and also being a huge antigenic target. And so I think it's just always under these strong evolutionary pressures.
Cool. And then we have one last question from Stephen Chen, who's asking, what's next? You focused a lot on SARS-CoV-2. Are you planning on focusing your career on studying interactions between human and viral proteins and its effect on pathogenicity? So I think this is another question that might help you make another call out to recruiting some students to your new lab.
Yeah. So SARS-CoV-2, I'm sure we'll still do some stuff in there. I don't have any burning questions about it right now, because its evolution has just gone kind of haywire now with all these Omicron subvariants. I mean, they're doing a lot of interesting convergent evolution of more antigenic mutations, but there'll always be interesting stuff there. But I am definitely kind of zooming out to other stuff. And so there's definitely some questions about the kind of broader Sarbecovirus evolution that I'm excited to get into, how these promiscuous pressures and perhaps antigenic selection in bats leads to a lot of this diversity that we see here. I'm excited to kind of extend these same approaches to other, families of viruses, especially coronaviruses. I've piloted yeast display of a bunch of these coronavirus RBDs, and they typically always express well. So there's also other kind of emerging and re-emerging coronaviruses like MERS, where I think kind of doing some of these same approaches a little bit more proactively before there's a global pandemic could be particularly helpful. There's also a lot of other coronaviruses that just, they're now common cold coronaviruses, or they haven't exhibited spillover, but they still have interesting dynamics going on. And so I think those will be a really fun, different groups of viruses to keep studying. And then actually, the reason I got into all this work was I was studying more of the antibody viral coevolution side. with the project I started my postdoc on, which was the development of HIV broadly neutralizing antibodies and how that cycles with viral diversification to prompt their development, kind of from this molecular evolution perspective, because those are some really cool proteins that have a ton of really cool clinical and, tons of biochemical data and things like that to synergize with the DMS. And so I'm excited to get some of those projects kind of rolling again after I got clearly very distracted.
Okay, great. Well, thanks again for the extremely impressive talk. It's like a very impressive and compelling body of work, and also timely. Thanks for taking the time to share it with us today. Thanks again.
Awesome.
So for everyone tuned in, we'll have our next seminar series next month, and we look forward to seeing you then. Thanks again.
Thanks.